and good afternoon to everyone. Okay. We are from Group 7 and this is our group member. We would like to present our mini lecture regarding to the glucose oxidase. Okay. That's why require us to describe the general information of the glucose oxidase. So, by using the Brenda website, we can get all this information. Using the Brenda website, we also can make the comparison of the glucose in the different organisms. Okay, for the application and market needs of the GOX, we can see first is in medical prospect, which is glucose biosensor for diabetes monitoring. As we know, diabetes patients have to monitor their blood glucose. So biosensor have use biocatalyst to undergo the process. So biocatalyst that we use is GOX. And in industrial food, in food and beverage additive, which is glucose oxidase, used to prolong the food and beverage by removing the residual glucose and oxygen. And third, in baking industry, which is GOX, have increased the dough consistency. And GOX also strengthen the bread matrix and improve the texture and quality of the baking product. And next.
Next is for task 3, the assay of GOX. This assay is used to measure the enzymatic activity of enzyme by fast spectrophotometric method using glucose as substrate. Also can use fructose and galactose. In this process, glucose oxidase will oxidize glucose to gluconic acid and produce hydrogen peroxide. And hydrogen peroxide used to reduce benzoquinone to hydroquinone. Okay, this is the procedure for this assay. The reason we choose this method because it is faster and easier. Okay, next is for task 4. The protocol of cell extraction from Aspergillus niger. First is isolate the Aspergillus niger in test tube. Then we add liquid nitrogen to allow it to evaporate and produce biomass. Then we add 5 ml of sodium citrate buffer and use for extraction of cell. Advantage for this method is simple and easy to use and can reduce time. Next.
Okay, this is the purifications of glucose oxide. This method is kind of general method and is kind of easy. Okay, this is another method to purify the glucose oxidase. Okay, there are 4 main steps and then have the sub-step. Okay, first is filtrations. The second is ammonium sulfate precipitations, followed by dialysis, and then the gel filtration using the Cephalus G75.
Next, the conformation of purified GOS. We can use the chromatography. The first is ion exchange with the separate by based on charge. We use this because to remove a target contaminant based on their charge. The next, the size exclusion chromatography, is we separate based on size and molecular weight, which is the larger protein will evaluate first, and we use the size exclusion because a short and well-defined separation which leads to good sensitivity based on their size and molecular weight. So next, the hydrophobic interaction chromatography, which is the separation based on hydrophobicity of protein, and we use this method because it's a good separation of molecule by differences of polarity. And furthermore, the affinity chromatography, because the separation based on specific biological interaction, and is used to remove contaminant based selective non-covalent interaction between an analyte specific molecules, and the reverse phase HPLC that use good for assessing target purity and the presence of contamination in the protein assay, a protein sample. Okay. Next, we can also use SDS page because it's very easy and rapid. It's very easy, rapid, and expensive. So the overloading the gel with 35 to 50 microgram of protein allows more sensitive detection of contaminating proteins. We also use the Western blot to identify and determination of intactness of target protein, which is easy to handle. And last but not least, the mass spectrometry is the best technique for establish protein identity and is to measure the presence of impurities with their mass.
Okay, so this is the 3D structure of glucose oxidase from Aspergillus niger. Okay, so we obtained this from PDB, protein data bank, and we view it through Yasara. Okay, so this why you use Yasara, because Yasara actually we can zoom each of the molecule or the residue. Okay. Okay, so to obtain the active site or binding site of an enzyme, we use UniProt and cluster W2 to determine the binding site. These bioinformatic tools can compare the amino acid sequence of different organisms. Okay, so the star level sequence is the highly candidates for the chosen active site or binding site of an enzyme.
Okay, next is task 8. Okay, what are the methods used to obtain the loss in activity? So our group choose X-ray crystallography because by using this we can obtain a 3D structure of the protein by crystallographer. They can obtain the 3D structure by measure the diffracted crystal. As we know, X-ray crystallography, they will blasting the crystal in diffracted differential way. So by measure the diffracted, we can obtain the 3D structure. By that, crystallographer also can obtain the 3D structure, the chemical bonding of the protein. So we can know what are the chemical bonding that causes the protein denature. Okay, so this is the pathway of the process X-ray crystallography, and this is maybe it's not too clear for you guys. So this is a simple overview of the X-ray crystallographic method. Okay, we put the protein and they will diffract it. Then computer graphic will obtain the 3D structure, and then we'll get the protein model. Okay, that's all from our group. Thank you.